Unravelling. I'm going to introduce the first speaker. Very pleased to have Joachim Surm here. He did his PhD in a far, far away land of Australia at the Queensland University of Technology under the supervision of Peter Prentice. And I believe you were working there on the animals you seem to love a lot, which are cnidarians. Is that correct?
Yeah, exactly.
And good. Joachim is also very interested in genomics and omics generally. So I think these two loves of cnidarians and omics intersect. I'm sure this will come out in his talk as well. He currently is a postdoc at the Department of Ecology, Evolution and Behavior at the Alexander Silberman Institute of Life Sciences at the Hebrew University of Jerusalem. And he is working there in the lab of Yehu Moran. So we are very pleased to have Joachim here. Today he's going to talk about a work titled Dominant Toxin Hypothesis, Unravelling the Venom Phenotype Across Micro and Macroevolution. So without further ado, Joachim, welcome. And you can take the lead and share your screen.
Thanks so much. Okay, great. Thank you so much for inviting me to present some of the work I've been doing these last few years. Yeah, as Ronald told you, I've been working on venom, specifically working on the venom evolution in sea anemones, sort of highlighted here in this picture. And I've been interested in venom for quite a long time, and what's always fascinated me in venom is using it as a model to understand evolution in general. More specifically is understanding the molecular evolution of venom. And when we investigate molecular evolution, one of the major pillars is gene duplication. Okay. So back in 1970, Susumu Ono sort of postulated and pioneered the idea that gene duplication has a major role in the evolution of organisms. So briefly, gene duplication is a process, a molecular process that involves the copy and paste of a genomic region. And this genomic region can be either the entire genome, the entire chromosome, fragments of a chromosome, or even just a single gene or fragments of a single gene. And following these gene duplication events, this can have very serious and important implications on the fitness of an organism. It can affect the morphology of an animal, the biochemistry of an animal, as well as leading to diseases and things like that. So following a duplication event, there are a few different evolutionary trajectories, such as pseudogenization, neofunctionalization, and dosage repetition. So in this really nice schematic by Casewell, we see that a physiological gene undergoes duplication, and then the duplicated gene can undergo this mutation and selection, resulting in pseudogenization, which is the loss of function. So there's an accumulation of deleterious mutations. The gene can also undergo fragmentation resulting from splicing, or these mutations can be beneficial and selection can act on it, leading to a protein with a new function, and this is known as neofunctionalization. And when we understand in venom evolution, neofunctionalization has been shown repeatedly to play a major role. Specifically, these physiological genes that can be found in non-venomous species undergo these duplication events and neofunctionalization to produce now a new protein with toxic activity. And that is then utilized by the venomous organism for things like predation and defense. Another fate of a duplication is this dosage repetition, or just amplification, where a copy can just be duplicated and now the organism has twice as much protein. Okay. So in many animals, venom is a complex mixture of molecules that can manipulate various physiological processes in the inflicted animal. And for venomous animals, many times the cocktail of venom is largely proteinaceous, meaning it's made of proteins and peptides that are encoded by the genome. And we can then consider venom as a complex trait or a polygenic trait, and understanding how these different genes that encode toxins affect the venom phenotype is critical in understanding the evolution of venom in general. One of the most striking observations we have on venom evolution is the convergence we see. So this review by Schendel, Jenner, and Untheim show that venom evolution has evolved convergently more than 100 times across 8 different phyla. And one of the earliest, or likely the earliest diverging animal that evolved venom was the cnidarian. So cnidarians consist of jellyfish, hydroids, corals, and sea anemones. And this is a phylum that is actually typified or defined by its venom system. So these animals all share a common cell type called a nematocyte. And this is a small cell type that injects venom using a harpoon-like structure. And although all cnidarians are considered venomous, sea anemones are arguably the most well-studied among cnidarians in terms of understanding their venom repertoire. And specifically, a number of different types of venom have been characterized in sea anemones, including neurotoxins that affect both sodium and potassium channels, pore-forming toxins, as well as proteases. And this is in large part due to work exploring the venom repertoire in the model sea anemone Nematostella vectensis, represented here. So Nematostella vectensis, very quickly, is the model sea anemone species. And it's a model system because it's able to complete its full life cycle in the lab. It's easy to maintain. It has a sequenced genome. It was the first sea anemone with a sequenced genome. We have many transcriptomics and proteomic data sets as well. And because of these, we also have a whole host of genetic tools that allow it to be manipulated, and as well as understanding the location of different genes. And because of these tools, we've been able to characterize a number of different toxin families. In particular, NV1 is considered the major component of the adult venom, highlighted here. And NV1 is a sodium channel modulating toxin that is produced in these ectodermal gland cells, not in the nematocytes. And what's really interesting about the NV1 toxin family is that it's found in a cluster in the genome with more than 10 copies that are highly, highly conserved. And although we've gained all these insights into venom and its evolution by exploring the lab strain of Nematostella, we are also gaining insights into population dynamics of venom by exploring the ecology of Nematostella. In particular, it's found in the Atlantic coast of North America, and it has very well-defined population structure, highlighted here. And previous work has shown that NV1 is very metabolically expensive, and during times of stress is massively downregulated. And there appears to be population-specific differences in how animals respond to stress and how they regulate the expression of NV1, highlighting that venom is having different population adaptations. We also have evidence that NV3, a paralog of NV1, has a restricted distribution found only in the Northeast. Taken all together, in this work, we wanted to understand how venom is evolving as a complex trait. So traditionally, many papers have explored venom in the terms of sequence evolution, but we wanted to understand what is the evolution of the genes that encode toxins and how this coordinated expression generates the venom cocktail and this venom phenotype. To understand the macroevolution of the venom phenotype. We then wanted to also further explore this by understanding how different populations evolve their venom phenotype, and if we see similar patterns at both different scales, at both the ecological and evolutionary scales. So to explore the macroevolution of the sea anemone venom phenotype, we first generated 29 de novo assembled transcriptomes from publicly available data. And from this, we made a species tree, and we quantified and mapped the expression of 8 different toxin families. From this mapping, we were able to generate the venom phenotype for each species highlighted at the tips of this tree. And an observation we saw was that for majority of the species, a single toxin family seems to dominate the overall venom phenotype, accounting for more than 50% of the total gene expression values, or TPM. We then wanted to understand how the venom phenotype has evolved across sea anemones. So we performed ancestral state reconstruction, and sort of quickly we see that the venom phenotype is maybe a bit more diverse, meaning that not a single toxin family is really dictating the venom phenotype. However, this should be taken in consideration that many of these values are not statistically significant. And I think this is likely the result of the fact that venom is, the toxin expression is evolving in a highly dynamic manner. And to test this, we modeled the expression of toxins and found that a mode of evolution of rapid pulses best explains the evolution of toxin expression in sea anemones, as opposed to early bursts or simple Brownian motion. This sort of supports that indeed toxin expression is evolving in a highly dynamic manner, likely to account for ecological interactions. We then wanted to understand what are these constraints acting on the combinations of venom components or toxin families and the toxin families themselves. So we performed phylogenetic covariance analysis. And from this phylogenetic covariance analysis, we found that indeed very little constraint is acting on these combinations of toxins, with the exception of NEP3 and NEP6, which are 2 toxin families found to be exclusively expressed in the nematocytes, hinting towards maybe some sort of synergy between these 2 different toxin families that have very different activity. We then could take these, this phylogenetic covariance analysis and plot using PCA analysis, or principal component analysis, to reconstruct the phylomorphospace of the sea anemone venom phenotype. When we look at this phylomorphospace, or this PCA analysis, first we see that the venom phenotype in sea anemones has relatively low dimensionality, meaning that only 2 components, or the first 2 components, explain more than 60% of the total variance. We also see strikingly that different species are clustering together that are distantly related, and that this clustering is largely due to a single toxin family. So we see that although the venom phenotype is comprised of many different toxin families, a single dominant toxin family dictates the venom phenotype, and that distantly related species can cluster together, or more closely together than closely related species, suggesting that some species are convergently evolving a similar dominant toxin. We then wanted to explore what is the genomic architecture of some of this dominant toxin families. We see that of these 8 different families, most of the species are clustering into 2 distinct toxin families, the NaTx family and the KTX3 family. And recently, there's been 3 genomes that have been generated using long-read sequencing. And this allows us to explore for the first time the genomic architecture of these dominant toxin families to a very high degree of specificity. So when we explore the genomes of these 3 different species, we see first that indeed the dominant toxin for Nematostella, that's NaTx, and for Actinium scalanthus is KTX3, that these two toxin families have indeed undergone massive duplication events. In fact, these toxin families, or the dominant toxin family, has undergone the greatest expansion compared to all other toxin families, highlighting that the dominant toxin family dictates the venom phenotype, and that gene duplication is largely driving family to become dominant. We also see that many of these copies are actually in a scaffold of tandemly duplicated sequences. And these tandemly duplicated sequences found within a cluster are often much more closely related to each other. Because we have chromosomal level assembly for two of these species, and we have long read sequencing, we can then explore the macrosynteny of these toxin families. So Actinia of the three different species share both NaTx and KTX3, whereas Nematostella only has NaTx and Scalanthus only has KTX3. We could then compare to see, is there any relationship or synteny shared between these toxin families? And what we see here in this Oxford plot on the right is that we see broadly that these three species share macrosyntenic relationship. However, when we explore these toxin families, we do not see any synteny, or we see no macrosynteny shared. However, when we explore the toxin families of other families, such as NEP3 and NEP6, we do see a relationship between some synteny shared between these different species, suggesting that they share some ancestral copy. This also suggests that the NaTx and KTX3 family, which are dominant, are evolving in a highly dynamic manner at the genomic level. Another interesting fact about NaTx and KTX3 is that they share a very similar cysteine framework, highlighted here in this web logo down the bottom. However, when we take these sequences and cluster them, they are still distinct. And because they share this similar cysteine framework, it hints that they may have a similar evolutionary history. However, when we looked at the synteny, we still couldn't resolve whether any of these shared some sort of homology. And this is likely suggesting that indeed this is evolving in a highly dynamic manner. So once we explored the macroevolution of sea anemone venom, we wanted to explore this at the microevolution scale, or ecological scale. So we looked into Nematostella vectensis across different populations. So we generated transcriptomes from 9 different populations, and found that these species cluster very strongly, have very strong population structure that are clustering largely to their geographic location, having clear northern and southern clustering. We also see that for the NaTx family in these species, NV1 and NV6 is shared among all populations. NV2 appears to be lost in Florida. NV3 is indeed restricted to the northeast. And NV4 and 5 and 7 have a more restricted or patchy distribution, likely due to it having a very specific temporal expression and only being expressed in adults that are female and likely have gametes. When we explore the allelic variation of from these transcriptomes to see the allelic variation of NV1, we see that most copies have many different paralogs, upwards of 8 for all of them, except for Florida, where Florida we were only able to find a single paralog of NV1. And this was very striking. However, from transcriptomics, we are unaware if this single copy is just in a high copy number or is in fact a single copy in the genome. So in collaboration with a lab in North Carolina, Ed Smith and Adam Reitzel, they performed qPCR using genomic data, and found that indeed Florida has a single copy in the diploid genome. They further show that there's significantly different copy numbers in different populations, except for Maine and New Hampshire, and suggesting that the both expansion and contraction of NV1 is highly dynamic. We also see that there's a positive relationship between the copy number of NV1 and the expression and its expression. We then wanted to explore whether what is happening at the protein level, because venom is acting at the protein level. We then performed comparative proteomics between two different populations, Florida and North Carolina. And we found that NV1 is the most differentially abundant protein between these two different populations, confirming that indeed the contraction or loss of copies of NV1 results in the almost total loss at the protein level compared to North Carolina. We then wanted to further explore the sequence diversity of NV1, and Jason, Ed, and Adam performed amplicon sequencing of NV1 at both the CDS and genomic level and found 30 different variants. When they performed hierarchical clustering, it shows that there's clearly 4 distinct core haplotypes. And besides variant 1 that is found in all 4 haplotypes, specific paralogs of NV1 are restricted to a given haplotype. To confirm this, we performed long-read sequencing for 4 different populations. In addition, there's the lab strain from Maryland. And this data perfectly corroborated the amplicon sequencing data, further supporting that certain paralogs of NV1 have exclusivity to a given haplotype. We also see from this data that indeed Florida has undergone a massive contraction, and that there is very little or almost likely no recombination occurring between different haplotypes. And this suggests that, that the gain or loss of these copies is occurring through some sort of duplication of the haplotype itself. And taken all together, we see that the venom phenotype in sea anemones is largely dictated by a single dominant toxin family, and that this dominant toxin family is likely selected or driven by a process of gene duplication. And that gene duplication can really act in a very dynamic manner, resulting in an expansion, even within ecological scales. And we also see a dynamic loss of copies. And given that NV1 is highly metabolically expensive, potentially this Florida population has lost its copy to account for this metabolic cost. We also see a striking similarity between the sea anemone venom phenotype and snake venom phenotype, in which they also show that a single toxin family largely dictates the snake venom phenotype, and that only a few of these toxin families are really the major families involved in their venom phenotype. And from this, it suggests that these distantly related organisms that have convergently evolved venom are evolving their venom repertoire using similar processes, likely due to gene duplication to drive a single toxin to become dominant. So altogether, this work is in collaboration with a great group from North Carolina. And we also had I had some help from some really talented undergraduate students. And I'd like to thank my lab and YooHoo for all their support these last few years, and our funding agencies.
Wonderful. Thank you, Joachim. That's great. If people have any questions, I can see Sebastian is complimenting Joachim on his facial hairstyle. I second that, Sebastian. Indeed, a nice talk. If people want to ask questions, Joachim, if you can unshare your screen, I would say just raise your hand. I will see if there's a hand up, so we can. If you're too shy to talk, then you can type it into the chat. I have a few questions if nobody else does. Let me see. No, I think you all fit on one screen. This is not a question because I was quickly looking in my copy of Sunagar and Moran, who, the paper that talks about ancient lineages having different venom compositions than younger lineages, and that venom composition needs time to stabilize or whatever, and what your results on a microevolutionary level say about that. But let's not talk about that. When you talked about downregulation of NV1 at some point in your talk in response to environmental stress, I was thinking, when you work on the animals in the lab, how representative are they of the animal in the wild? Because in the lab, if I don't misunderstand, they are pampered, gigantic, fat little babies. Well, they don't have adipose tissue. If they would have, they would be fat, but very unstressed, I imagine, compared to their species cousins in the wild. If you compare a representative venom profile of a lab Nematostella with one from anywhere in its distribution, are they pretty representative?
That's a great question. So we have some pilot studies where we explore actually samples taken from the wild and not put in RNA later straight away, and we have transcriptomes from those different populations, and we still see that NV1 is really the major toxin, and it's still very highly expressed. But it's a very good question. In our lab, they are obviously really fed like ideal conditions. And given that it is so metabolically expensive, it would suggest that they are able to accumulate this in massive amounts. But I guess also they're not really ever introduced to like native predator prey. So you could also argue that maybe they would lose some of their expression in the lab. But we do see for venom to date, very good representation of the venom for the lab and both the wild population.
Yeah, I mean, what you just said there, you basically talk about the topic of venom plasticity, where within a given genotype, you can have different expression profiles based on environmental effects and interaction with the environment, which is actually the world of venom, a really poorly studied topic, because in snakes, there is less than a handful of documented cases where one toxin may be more or less absent when you keep the snakes in the lab after you catch them in the wild for 15,000 years, and minor, mostly very stable. So systems that seem to be so responsive to the environment, like these cnidarians, I'm so happy that this research is being done, because I have a suspicion that we miss an entire bit of venom variation from ecological time to shallow evolutionary time. So I'm very happy that you guys do this kind of research. Any people with questions, speak up. I'm going to turn my lights on because thunderstorms are coming into London. Maybe one more question before we hand over to the next speaker. Again, NV1, a venom toxin that is not injected. So typically, is it the venom then? It's applied by glandular cells on the surface of prey. So these peptides must be absorbed remarkably quickly. Are there other examples in other groups of venomous animals that you know of where neurotoxins can be applied to the outside to have an immediate effect?
That's a very good question. I don't know if we know the exact mode of action as to how NV1 is getting into like the predator or prey. They are ectodermal, so they are, I guess, housed close to like the nematocyte. Regarding other animals, whether they have something similar, maybe you would know better than me, but is there like maybe some toad or there's some frogs that have like, I guess like gland cells on their top? I guess it's maybe more indicative of poison or something, but if they get ruptured, they can maybe release it. So we know.
I think that, vaguely, I would have to look it up. I think some work that Kim Roelands in Belgium is doing with some frogs, where some peptides seem to open up crevices in the epithelium quite quickly. And so basically they create an opening to get in between cells themselves. I think that's another interesting, because the field of research, you know, the definitions we have for discontinuums in nature, poison, venom, and then there is this gray area, and it's interesting to talk about that over a beer. But since there is no beers here, Joachim, thank you for that. I see there's no further questions. Thanks very much.